From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 47, recorded on January 22nd, 2024. I'm Vincent Racaniello, and you're listening to the podcast about the nervous system. Joining me today from Salt Lake City, Jason Shepard.
Hi, Vincent. Hey, Tim.
And from New York, Tim Chung.
Hello. Hi, everyone. Nice sunny New York.
Tim will tell you that it's really cold out, isn't it?
It's really cold. And then when I go running, everything's iced over. So I have a tendency of slipping. Hopefully don't suffer a spinal cord injury.
I hope you don't, because this guy had an injury on his bicycle, apparently.
Mm.
Yeah, we've actually been pretty warm here after the massive amount of snow that we got.
Oh, yeah, that's good. That's good. Yeah, this has been unseasonably cold, and it's going to continue for a long time.
Mm. It's good.
Yeah,
it's finally
keeps you inside.
Kind of feels like winter.
It does feel like when there is actually some snow around too. So we had 690 days without snow, and then suddenly we had a few days of snow. But there's like an inch at most.
Yeah.
And here in New York, there's none. You have to leave the city to find. Maybe Central Park. You haven't been to Central Park, have you, Tim?
Not really, but like in my local park on the East River, there's some snow, but it's mainly melted and refrozen into ice, which is annoying, which is hazardous.
Today we have an amazing paper, which we kind of gave you a hint of in our chat, and Tim is going to walk us through it. What do you think of that? Walk us through it.
That's a very good segue. So today we are covering this paper that was published last year, actually, and I remember it. I think that's quite a lot of publicity that went with it, and rightly so, because it is an amazing paper. And the paper is titled Walking Naturally After Spinal Cord Injury Using a Brain-Spine Interface. And it was published in Nature 2023. And the first authors I think there are two co-first authors. They are from Switzerland, Lausanne in Switzerland. So things might be French. First author is Henri Laurard and Andrea Galvez. I hope I pronounce those names correctly. And then there are three senior authors, William Charvet, Jocelyn Bloch, and Gregoire Courtine. My apologies for pronunciation. I try my best. So this paper that we're going to talk about today concerns a single patient. So it's a case study. And the patient has had a spinal cord injury. Let me check where. Let's see. The patient had a spinal cord injury that is incomplete. So the spinal cord isn't severed, but it is injured at the cervical level. Apparently, that's the most common injury for spinal cord is you fall and you kind of break your neck and cause an injury to the spinal cord. During a biking accident 10 years before. And this left him paralyzed. It seems to be at least in the legs. So he seemed like, I should mention the paper is open access. And the paper has, so it will be on the show note, and it has 4 accompanying videos that pretty much summarizes the paper, including all the technology. And you can also see how the patient is recovering. So I encourage everyone to go and check it out because it really is quite amazing. So the patient has, even though the spinal cord is injured, he seems to have retained some of the upper limb mobility. So he can hold himself up using a walker and also using crutches. But the lower legs are almost completely paralyzed. So he cannot hold his own weight. He can't move them. He can't walk at all.
So, Tim, can I ask you, so is that because the spinal cord was not fully severed? There's still some connections for his arms and respiration and all that?
That's correct.
Okay.
Absolutely. So if it's completely severed at the cervical level, I'm guessing then you're
Q4 then, right? Yeah.
Yeah, you can't. I'm guessing you can't breathe. Yeah, but the lower limbs are definitely gone, and not often talked about in spinal cord injury, but also affected are things like autonomic function. So like bowel control can be affected, and also like sexual dysfunction is also a huge concern. So there are many things that needs fixing. But today we're going to concern with just the motor side of things. So I think before we get into the paper, we should have a little bit, we need to talk a little bit about how in normal, uninjured vertebrates, let's say, how walking is achieved. Just so that we know what kind of the engineering that is going to be involved in today's paper. Basically, very brief summary of today's paper is the research team is going to figure out a way to stimulate the spinal cord using some electrodes that is implanted to basically artificially reconstruct walking by activating the correct muscles at the right time. But those activation will be controlled wirelessly by the brain. So the patient would think that I want to take a left step, take a right step, take a left step. And then that would cause a stimulation in the spine to trigger the correct muscles to allow him to walk. It is quite an amazing feat. But as background, we're going to talk a little bit briefly about how walking is achieved in healthy vertebrates, at least. So a quick quiz for you guys. How much of the brain do you think is needed for you to walk, for you to be able to walk? Do you even need your head?
Well, there's been, the basic locomotion, you just need the spinal cord. But for controlling it, you need the motor cortex, right?
Turns out you don't actually need that. So so have you heard of this thing called headless chicken? Running around like a headless chicken.
Well, that's what I thought. Yeah, you just to walk, you don't need the cortex. But I thought to control it.
It turns out you actually don't even need the cortex. So one thing when I was just doing some research for this paper, I looked up headless chicken, and as it turned out, there was an actual headless chicken historically called. People should look this up. It's called Mike the Headless Chicken. And if you want to find something to gross your friend out, you can search for it online. There are pictures of Mike the Headless Chicken. It's a chicken that was supposed to be someone's dinner, but the person who was decapitating it did a poor job. So it cut off most of the head except the brainstem. So the brainstem is still there, but pretty much the whole head has come off. And somehow the chicken didn't bleed to death because there's a blood clot. And it was able to run around normally. And it was kept alive for a whole year after the head came off. A whole year. It was fed through like a pipette dropper, putting in food directly into the esophagus. And the poor chicken, not only did it survive a botch decapitation, it was then kind of carried around as like a sideshow circus for the owners for a year. So that was an actual.
I presume they took the head off at some point, right?
Oh, they I think they carried the head around as part of the show. So the head was off. So yeah, the head is completely severed with a beak. But the thing that remained was this thing called the brainstem, which is kind of past your. So from the front of the brain to the back of the brain, you have your cortex, which is, as human, most of our brain. And then you have things like the basal ganglia, the thalamus. If you cut all those off, and if you cut all those off, and perhaps the cerebellum, what you're left with is brainstem, which consists of like midbrain, things that control your breathing, things that control maybe whether you're awake or not, whether you're conscious, and as it turns out, whether you can walk. So much of these walking experiments, so that was a botched experiment from the 1950s. But as it turns out, a lot of walking experiment was done, I'm sorry to say this, it was done about 100 years ago, like during the early 1900s. So it was done using cats. And these cats would, the researchers would look at surgically cutting the cat's brain at different places, at different locations, to see how much of the brain they can disconnect from the rest of the spinal cord for the cat to still be able to walk. And what they found is they can basically have this preparation, which is called the decerebrate cat. So you basically cut below the thalamus, and all you have is the midbrain, the brainstem and the spinal cord. So the cat cannot see, it cannot hear. It's basically almost unresponsive to the external environment. However, if you put this cat into a harness, so you support its weight, and underneath this cat, you put a moving treadmill, funnily enough, the cat, even though it will not respond to any external stimulus, it would adjust its step to the treadmill, just like a normal cat would. And if you increase the speed of the treadmill, the cat would go from walking to start running to like full-blown galloping. So like when all the four paws are like off the ground and running around. So it can even transition its different state of moving from just gentle walking all the way to full-blown running. So it suggests that a lot of the kind of the machinery, the system for controlling walking, or for vertebrate in general, in neuroscience, we call it locomotion. Just means you move around, you carry your whole body around. It actually probably involves just the brainstem and the spinal cord.
But Tim, the chicken and the cat, they don't think any longer. They have no cognition at all, right?
Yep. As far as we can tell, because the brain is gone, the cat is, if there's any consciousness, it's not a sort of consciousness that we can identify with. Like
amazing.
Yeah.
Wow.
Yeah. So I mean, I guess, but this is has been well established that the brainstem plus the spinal cord are sufficient for the motor loops and the sort of reflexes that control walking. But of course, you have the motor neurons coming from the cortex, which are the longest neurons in the body. They're still controlling the top down, whether you're running, whether you're walking, but of course you can't learn different motor things without the motor cortex. But yeah, it is I think well established that you don't need it for just being able to.
Yeah. So I think the idea, the theory is that locomotion, just plain walking, is such a kind of almost basic or like evolutionary conserved skill that almost all creatures that has to move around to look for food, they need to do.
Right.
And because of that, it kind of a lot of the machinery for computing what I need to do, move my legs, so move my left leg, then my right leg, that kind of stuff, has been kind of outsourced to the spinal cord. And what the brainstem is doing is basically selecting the correct spinal cord program so that you can change your speed if you need to. And then what the motor cortex is needed for is for things that are a little bit more intricate, like picking up food, handling food. For example, like if you have some obstacle, as you're walking, if there's an obstacle, then you start seeing your motor cortex fire so that you can adjust your gait to step over the obstacle without tripping over, that kind of stuff. So, as it turns out, people have been looking for these kind of automatic rhythmic firing of the spinal cord that looks like you're moving in the spinal cord for a while. And these kind of rhythmic neuronal firing that might mimic walking is called central pattern generator. So as far as I can tell, the one definition of a central pattern generator is basically a system, like a bunch of neurons, that can generate rhythmic activity patterns without a rhythmic input. So it kind of intrinsically has some connectivity that allows it to, for example, when you're walking, your left spinal cord neurons for your left leg would fire for a little bit, and then it would stop firing, and your right leg motor neuron would fire, and then you alternate between the two. And this is mainly studied actually in a interesting way. It's studied most studied into the lamprey system. So these are like jawless fish that don't even have legs, but for them to move around, they do have to kind of undulate left and right. And people have found that if you isolate the spine, so just the spine, even without the brainstem, and if you put in the suitable drug cocktail onto the spinal preparation and then record from the spinal cord, you see a very characteristic left side firing and then the right side firing, and then the timing from the top of the spinal cord to the bottom of the spinal cord is also coordinated. So it kind of, you can almost, they call it a fictive locomotion. So it's kind of brain in a vat, but it's spinal cord in a vat, matrix style.
Yeah, and these intrinsic rhythms are highly conserved, even all the way to like invertebrates, like leeches, and there's also this really important circuit in crabs, and so there's the ability of neurons in a circuit to control timing and rhythm is one of the key evolutionary adaptations that they.
And in fact,
do.
Okay. In fact, one of maybe the most famous and perhaps most vital central pattern generator in us is our heart, because that.
And breathing.
And breathing, that's true. Yeah, both of those go hand in hand. So for the heart, it is basically you have a cardiac pacemaker that generates its own rhythm to allow your muscles to contract. And when that fails, doctors and engineers have come up with artificial pacemakers that you can plug into your heart, and it would generate stimulation, the appropriate stimulation to get your heart to contract. So that is kind of the idea for what today's paper is going to be. The connection from the brain or the brainstem to the spinal cord has been severed. And can we do something to the spinal cord to trigger walking again, kind of artificially? So this is definitely the holy grail of spinal cord injury. Although I should quickly mention, another thing that people are also looking into is, can we cause either the damaged neurons to regenerate or the remaining neuron to grow more to take up some of the slack of the degenerated or injured neurons. So there's also there's a completely separate molecular story of how can you put in the right growth factors. And I think back in TWiN 34, a few episodes ago, Vivienne talked about how important microglia are in kind of limiting spinal cord injury to make sure the injury is not as bad as, doesn't worsen even worse than it can get. So there's definitely a separate kind of track that we can look at for spinal cord injury. But one engineering solution is to basically...
But it hasn't been very successful. I mean, I think there was a lot of, even a decade ago, there was this idea of using stem cells to regrow neurons, and then that failed partly because you can implant cells, but they wouldn't know where to connect to. So it's reforming the right connections has been the biggest challenge. So you can get regrowth, you can get put in cells like stem cells, but that's been the challenge. And the thing is, like, other animals do it, so even fish do it. So it's sort of remained a promise that once we understood what the factors are to control the wiring, we could do this, but nothing's worked so far. Really.
I think one downside, so we're going to get into this, but one downside is how to translate, not let alone fish to humans, but just like, mammals to human can be difficult. Mm-hmm. So inspired by this, one of the authors of the paper, Gregoire Courtine, in some of his previous publication, he's looked into this and he used a rat model. And I remember this was published like 10 years ago. I saw some of the videos and it was like amazing. So he had a spinal cord injured rat with an injury so severe that the hind legs, the the bottom legs are completely not moving, paralyzed and flaccid. And he found that by stimulating the spinal cord in the lumbar region, so where your legs are, and also by putting in the appropriate chemicals, so these are monoamines such as serotonin and dopamine, to encourage the cells in the spinal cord to fire, perhaps endogenously. So by this kind of combination of electric stimulation and chemical stimulation, he can cause the rat to walk again on a treadmill, just like a decerebrate cat did. So remember, the decerebrate cat had no cortex, but the connection from the brainstem to the spinal cord was still intact. So the cat could adjust. It can feel, not consciously, but the brainstem and the whole system can feel, in quotes, the treadmill moving and adjust accordingly. So the TWiN lab found that when you put in this chemical plus electrical stimulation in a rat, you can kind of recapitulate this responsiveness to treadmill, so the rat can step again, and that was hugely amazing. What's even more was that with kind of hardcore training of the rat, with a lot of this stimulation, kind of electric chemical stimulation, hardcore training, which is basically rehab, they can get the rat not just to respond to a treadmill, but to kind of, under its own volition, under its own control, to move its leg to get some food. So they would put a rat, so this took months of training of like rehab, rat rehab. They would have a rat a little bit far away, and then they have a bit of food, and then the rat would kind of, on its own accord, it is still weight supported, but at least it can move its leg to approach the food that is kind of held in front of it. And this was part of the inspiration for human spinal cord injury patients, because rehab, it's known to be a hugely significant part of kind of the recovery from spinal cord injury. And what people have hypothesized is that during rehab, some of your remaining residual neurons, at least for the incomplete spinal cord injury case, where there's some residual neurons that are still there, some of the axons are still there, but they were not, because they weren't used for walking before, they're not really by themselves able to trigger the spinal cord program to get you to start walking again. But with rehab, you might be able to get these axons to maybe at least have more terminals in the spinal cord. So just like if you like people with an amputation of an arm, you can see in the sensory cortex, what used to represent that arm gets invaded by other parts of the body. You can imagine some of these kind of adaptation to also happen. So the idea is that in rat, we have shown that we can do it. We can train a rat with rehab, and then plus this stimulation, the rat can recover a little bit of volitional movement. Can we do this in human? So I think the TWiN lab has tried very hard to get the same thing to work. And I think they found that the translation is not quite as easy as you would expect. So this might make sense, because if you think about it, humans and rats are very different. Rats are actually four-legged animals, and humans are bipedal. And walking in human is much, much harder than walking on all fours, like crawling. And human and animals are also quite different. For example, like things like gazelle and horses, like once they're born, within like a few minutes, they shake themselves up and then they start galloping away. Whereas in human, it takes like a year, or I'm guessing around a year, for a baby to learn to walk and learn to balance. So walking is a much more challenging endeavor, and it is actually unknown. So I don't think there are kind of experiments on humans like this, but like a decerebrate human on a treadmill, can they walk? Like highly unethical. I don't know if it's doable. Although there are also reports, I think, that there are some babies born that doesn't have, due to some health reasons, they don't have most of the brain. But if you move the legs on like a flat surface, kind of like a treadmill, they start stepping as well. So maybe some of the central pattern generator in the spinal cord still exists in human. In any case,
okay, so let's get to the.
Sorry, that's
the first figure.
So we're going to we will go to the figures, but for
for the paper, what the TWiN lab actually did is they spent years to find the right stimulation for the spinal cord so that you can actually walk again. So as it happens, in humans, they actually didn't end up doing the injection with chemicals. I don't know why, but maybe the chemicals don't work as well in humans, or maybe there are concerns about infection. But with humans, they took the approach of just stimulating with electric pulses. So this is the kind of pulse stimulation that people use for deep brain stimulation in Parkinson's disease. But it is a array of electrode that they insert epidurally into the spinal cord. So this is above the dura of the spinal cord. So they're not directly touching the spinal cord, but they can basically emit electric field that would cause the spinal cord to fire. And there's certain interesting consideration going on here, which is the easiest place to access in your spinal cord is from your back. But as we talked about previously, the bit of the spinal cord that has all your sensory neurons are the dorsal side, like pain in your back, the mnemonic. So they insert the stimulator onto the dorsal side of the spinal cord, but all the motor neurons are on the ventral side, and those are the motor neurons that you want to get into. So it is, I'm guessing, technically quite challenging to get those electrodes to go to the ventral side without cutting all the nerves that come out of it. Like topologically, it's very difficult. So engineers are stuck on the dorsal side of the spinal cord, and people have to try to somehow activate the motor neurons on the ventral side. So
the dorsal is our back, right?
It's like dorsal fin of a dolphin, like it's on the back.
Got it.
And ventral is the stomach side.
Belly.
I have no good way of remembering what ventral means, but it's the stomach side. So what the TWiN lab in the previous paper, what they found, and this is exactly what they did in this paper as well, is they found a very, very nice way of stimulating the correct muscle. So in humans, as it turns out, we are as yet unable to find a way of triggering a central pattern generator that would immediately cause all your muscle to automatically walk around. So in lieu of that, because they can't do it, what they did was they had a healthy control. This is not in this paper, I'm just still background. The paper would come soon, I promise. But this is important because it tells you how they came up with the stimulation. They had a healthy control, and they put electromyograph detector, which is EMG. It detects when your muscles are firing. They put it all over all the leg muscles and asked a healthy person to walk. And they recorded exactly which muscle is contracting at which point of the walking cycle, gait cycle. Okay, so they now know what they want to what kind of muscle contraction they want to actually recapitulate in the spinal cord injured patient. Next, as it happens, a lot of surgeons and neuroscientists have mapped out so you know your spinal cord have different segments going from the top of your neck all the way down to the bottom of your spine. They have different segments. As it happens, the motor neuron that innervates different muscles in your leg, they kind of are segregated at different segments. So what they could do is by selecting the electrode at different segment of the spinal cord, they can try to target different muscle group using that way. And how they can actually get from the dorsal side of the spinal cord to the ventral side of the spinal cord where the motor neurons are, is they stimulate these kind of nerves called dorsal root entry zone nerves. So these are basically the dorsal root nerves that carry sensory signal, like what we're talking about. This is like all the pain nerves, all the proprioceptive nerve of where your joints are. They stimulate those neurons, those nerves, and those nerves indirectly actually talk to your ventral motor neurons on the ventral side. So this is like one of the reasons why you have things like reflex. When you tap under your knee, your leg kicks, and it's because your sensory nerve goes to the spinal cord and it talks to the motor nerve on the ventral side, and then that causes your knee to contract.
Or heat or pain sensory, the same thing. Right?
Exactly. So this is very much like a few episodes ago, we talked about electroacupuncture, where we want to shut down the immune system by stimulating the vagal nerve. The vagal nerve is really deep inside us. It's hard to stimulate it. Well, we just jab a needle in our sciatic nerve, and that somehow travels all the way up to the brain and then indirectly stimulate the vagus nerve. So they stimulate these dorsal root nerves, and that indirectly stimulate the correct muscle on the ventral side. So if we have time later, immediately you think about, are there side effects when you stimulate these sensory nerves? So we can talk about that if we have time later on. But this is how they're able to reconstruct. They can now stimulate different segments of the spinal cord, and then by doing that, have correct group of muscles activate at the right time, at the right place to reconstruct the muscles to step. So this is huge engineering feat. You are almost like designing a robot and getting it to like contract correctly. So we don't have a central pattern generator yet that would do it automatically. So this they did previously, but what they found was that they had the patient undergo this stimulation and go through rehab for a very long time, for almost 3 years. And the patient got better, definitely got better. So it could actually go from completely unable to even stand up to being able to stand up when it's stimulated, when the patient's stimulated. And with rehab, the patient, you can turn off the stimulation to the patient, and the patient can remain standing and take some small steps on a flat surface. So it can actually, this is incredible. The patient can move a little bit without the stimulation. So we think the hypothesis is that some of this kind of post-injury adaptation of maybe the terminal sprouting to take over some of the kind of slack caused by the injury is happening. But as it happens, the patient reached a plateau, so he wasn't improving very well. And part of the complaint is that the system that they previously designed, it actually sensed a little bit of for the patient to start moving again during the stimulation, it sends a little bit of residual movement the patient had in its heel. Like even though the patient had almost complete paralysis, its muscle can twitch a little bit at the heel. And I might have read this incorrectly, I apologize if it's wrong, but from what I can understand, when the patient twitched its heel, it would cause a stimulation and would trigger walking. But the downside is that was very kind of, there's a lot of delay between the patient's intention and walking and the walking. And it's kind of like if you give a talk like in a conference room or you are on a Zoom meeting and you can hear yourself, but with a feedback, it is very disruptive. And it's a little bit like that. You can make, like, it caused walking to be quite difficult. So finally, we get to this paper, which is they try to solve all this engineering problem. So what they decided to do, what these doctors decided to do is instead of having the patient kind of manually trigger his heel to initiate a gait cycle, which can be very disruptive, they actually measured the patient's brain waves and try to find a brain wave that correlates to when the patient wants to move and use that to trigger the stimulation. All right, so finally, we're on to figure one. What they did was they designed these electrodes that would fit onto the motor cortex of the patient. And these are surface electrodes, and they measure the brain waves. And as it turns out, the motor neurons that control different parts of your body are spatially distributed at kind of characteristic places on your motor cortex. So they basically, if you want to move your hip versus if you want to move your arm, different places of your motor cortex would fire. And because of...
So Tim, this goes back to my original point, which is that this is what you need the motor cortex for.
That's true.
That's what I was trying to
that's absolutely
bring
true.
it up. So, and the other thing that's sort of interesting here is that those cortical implants are not simple, right? That these are not surface as in on top of the skull. They're implanted in the motor cortex. So this is still very invasive. You still have to do brain surgery. You have to open the skull. This is the kind of thing that Neuralinks is saying that they want to do as well. This is Elon Musk's company. And a lot of what that company has been doing is refining the electrodes that you put in so that you can get, as dense an array as you can. So that's, so they're coupling this sort of new technology that's been, developed in academia, where we've done this in monkeys and whatnot, and now finally doing this in humans. But this is not the first time.
No, it's not the first time. Definitely not the first time, but potentially the first time in a spinal cord injury person.
Yeah.
Because I think people are also doing things like that for people with paralysis, so that they can type or like do some sort of handwritten style things. But for spinal cord, they just measuring essentially the brain waves over a wide area of the motor cortex. But they have enough electrodes so they can tell which bit of the cortex is emitting these different brain waves. So as it turns out, when you think about, when you intend to move, your limbs or your hip, or when you actually carry out the movement, different bits of your motor cortex would fire, and in a different characteristic brainwave kind of shape. So they leveraged this. They implanted these electrodes onto the patient, and then these electrodes are actually normally inactive because they don't have power. But if the patient wants to use these electrodes, this is incredible. What he does is he puts on these headset, kind of like big over the earphones. But instead of putting the earphones over your ears, the kind of the 2 cup goes above these implants, and the implants are implanted under the skin. They are kind of like replace bit of the skull. They're completely implanted under the skin, so you can't see it normally. If you see some of the videos that is open access online, he puts a headset above the implant, and the headset would both power wirelessly the electrodes and also record what the patient is thinking. And they would communicate to a laptop, and the laptop would decode what the patient is thinking. And based on what the patient is thinking, it would wirelessly talk to the implanted stimulator in the spinal cord to trigger the correct pattern of stimulation to activate the correct group of muscles for whatever the patient wants to do. And for the experiment that they did here, what they first did was, once these are implanted and the patient's recovered, they gave the patient kind of like almost like a game where they asked the patient to move a different part of the lower limb, at least think about moving. The patient can't really do it because he's paralyzed, but think about moving your right hip or your left hip, or moving your knee or moving your ankle. So those are the main joints you need to move in order for you to run. So those are the 3 things that you need to do. So the patient isn't actually moving it, although since he actually underwent a previous clinical trial, he might have been able to move it a little bit. But the patient is, watching on screen these cues, asking him to move this thing, to move the right hip, move the left hip. The electrodes on the brain are recording all the brain pattern that are associated with these different commands. And they try to pattern match. They kind of find a way of separating, clustering these different pattern of signal out using machine learning, so that you can calibrate this brain-spinal interface, this detector. So once it's done that, in the future, whenever the patient thinks, I want to move my right hip, it would get decoded by the implant correctly. So they did that. They asked the patient to, think about all these things, trained an algorithm to basically decode all this. And then, based on the previous study, they know exactly, if the patient wants to move the right hip so that it brings its knee up, they know exactly which muscles to activate, your hip muscle and a little bit maybe of mainly your hip muscle. Whereas if you ask the patient to like extend your knee, you need to activate your quad, that kind of stuff. So they have all these libraries of stimulation, and then couple it to the patient's thought, the patient's kind of thought command. And they showed.
Tim, what's the delay between the thinking and the actual movement, you know?
They say, I need to check, if I remember correctly, 100 millisecond comes to mind, which is actually quite slow. So if you, so don't quote me on that, I'll have to find it, but I think I read that it's about 50 to 100, it's about 100 milliseconds.
It's a perceptible delay. Yeah.
It's a perceptible delay. There's a lag. So for example, if I reach for my cup, I can do it kind of within the blink of an eye, which is maybe 50 millisecond. So there's a slight delay, but it's a lot better than the previous system, which has a much, much longer delay. So it's still, it's definitely
100 millisecond delay. Yeah.
So the delay has been shortened, even though it's still there. So there's still work to be done. But they show that they can actually get the patient to do this. They can ask the patient to move this hip, this joint, and then the decoder would be able to decode accurately, and then the patient would be able to move those muscles, or at least the stimulator plus the patient would be able to move those muscles appropriately.
By the way, just for comparison, audio delay, when it hits about 10 milliseconds, you can't handle it anymore.
10 millisecond?
Yeah.
Wow.
Yeah. So if we have an echo, 10 milliseconds, we're like, hey, there's an echo. I can't handle it.
Wow. Okay. Yeah. I mean, that's when you get into like those two people keep talking at the same time situation. Yeah. So I think there's potentially still work to be done, but it's the patient, like at the end, there's some questionnaire that they asked the patient, and the patient said it's definitely better than the previous system.
So what about this processing unit? Is that something the patient can carry with them?
Yes. So the whole thing is put in a backpack. So like the patient wears a backpack that supplies the power to the headset and also all the program for decoding and then how to stimulate. It's all in a backpack. So you can see a patient actually walking by himself, like on crutches. That's a second bid. I can't remember which figure we're on, but the first thing they did was they showed that they can calibrate the brain-spine interface so that you can independently move different muscles. And once you can do that, they can now use the program that they previously established to get the patient to walk again. So now the patient, instead of thinking individual joint movement, the program has a different mode, which is walking mode. As far as I can tell, I don't have the full documentation of the program, but it seems like if it's in walking mode, the patient just think, does he want to lift his left leg or right leg? So left or right, or rest. So like not doing anything at all. So as it turns out, the after calibration, the machine can do that perfectly well. And you can, so they, in the paper, they showed, the whole brain wave at different, when the patient is doing left walk versus right walk. It shows that the decoding machinery is able to accurately decode the left from right, and also when the patient is voluntarily resting. And they also showed that, and then when it's stimulated, that the ankle is actually moving along. So the patient is actually now on crutches and he can walk. And if you turn off the stimulator, like halfway through without telling the patient, you can see that the machine still decodes that the patient's thinking go left, go right, but the muscle activity is drastically reduced. Which is wonderful. And not only that, they also put like sensors on the different joint, motion capture sensors, to see how the trajectory of the kind of like the joint angle as the person is walking, how the whole leg works. This is called kinematics. How the kind of like the whole physics of the walking movement for the patients, how is that compared to a healthy control? And how is that compared to the previous kind of clinical trial where the patient had to initiate movement kind of haphazardly using like a muscle twitch? And they show that using this brain-spine interface, it does a much, much better job it's not identical to healthy. And if you watch a video, even though the title said natural walking, it does not look at all like an uninjured person walking at all. However, it is walking at least, not needing its weight supported. The patient now can support his own weight using his legs and using some crutches. So on that aspect, it is natural, and also like moving left, right.
Why are the crutches needed? Because he doesn't have the strength yet, or
I don't know for sure, but one thing that you can see in the video is that I think the full gamut of the balance that is required.
Yeah, I think it's the balance, because the problem with the balance is that you also need that really instant proprioception feedback to say, oh, I'm not balanced correctly.
Yeah.
That's probably really difficult to
That is super difficult. And this is one of the side effects that we said we might get to, is that they are stimulating in order to pull the strings as like a puppet master of your different muscles. They have to stimulate the dorsal root, which contain all the proprioception. So the same group actually showed that if you just go stimulate willy-nilly, it actually really disrupt walking, because the proprioception signal is completely disrupted. Actually, there's a very rare, run out of time, I have to be fast, but very quickly, very interesting thing is there's a very rare clinical case of a few patients in the world where they're infected by a virus, and this virus selectively destroys the proprioceptive receptor, or the neurons that carry proprioception. And these patients cannot walk. Like, they literally have to crawl around, even though they can move all their body. So it's kind of like the opposite of this case. They have their muscle function. They can like pick up things if they want to, but they have zero sense of where they are, where the joints are. It's kind of like if you get very, very drunk at night and somehow you're driving, don't do that, and you get pulled over by the cop and they ask you to close your eyes and then touch your nose. That's a thing that you lose when you're very drunk. And when that happens, and it's like people think it's caused by a virus infection. And when that happens, patients cannot walk. And there's one case of patient that retains some walking ability purely by visual cue. So he like looked very hard where he should put the foot and like how far he should, like the next step should be. Anyway, sidetrack. People can try and look it up. There's a BBC documentary on this. Back to the paper, using this kind of brain-spine interface that really speed up the time delay between thought and movement, and also how natural the whole thing is. The patient can actually, it shows in the paper that the patient can use a little walker, like those are like the little like trolley with wheels that the patient can use, that you see sometime kind of old people also use to help with the balance. The patient can use that and go up and down ramps, which the patient was never able to do before. The patient can even walk on stairs, which, I mean, that's incredible for a paralyzed patient. And also on like completely uneven surfaces. So presumably the decoder can actually decode the amplitude as well. So you can, as far as I can tell, it can decode not just which joint you want to move, but also how far you want the joint to move. So you have some of that control. So the very final point of the paper, the very last figure, is that this stimulation is all well and good, that we can now walk more naturally. But one of the main things for spinal cord injury is rehab. And you want to somehow trigger some of the remaining nerve to kind of take up some of the slack to help you walk. So what they showed is that this patient, when they're undergoing, so when they did the previous stimulation, the patient reached a plateau despite intensive rehab. However, with this new stimulation that kind of really reduced the time between thought and movement, the patient actually improved much further from the previous plateau. And all this is conducted after rehab, and then they can turn off the stimulation and show that the patient's improvement actually shot up. Which is wonderful, but there's a potentially interesting mechanism going on, which is what is going on during rehab, presumably, is that the patient is using its brain to tell its limb muscles to move again. And there's a brain signal that is coming down from the residual kind of still uninjured neurons, coming down to the injured spinal cord, to your lumbar leg region, and desperately trying to talk to whatever neurons is still there. But without the stimulation, this communication doesn't work very well. But with the spinal cord stimulation, then your brain signal and your leg signal can now communicate at the right time. And there's quite possibility some sort of plasticity going on that requires the timing to be timed perfectly. There are things like phenomenon that we didn't talk too much about here, called on the podcast, called spike timing dependent plasticity, where the timing of the presynaptic and postsynaptic neurons have to be really exquisitely tuned for the plasticity to occur. So maybe by improving this timing between the brain signal and also the leg signal by stimulation, you can get much better rehab going on.
This is actually where I think the chemical side, the molecular side, can converge. Where we're trying to figure out how to boost plasticity in a way that's sort of permissive to wiring, and this is where I think in tandem with sort of more of the engineering side of things could promote recovery.
Yeah. That's what I was asking, whether neurotrophins, could be combined with this, right?
Yeah.
That's definitely another side of all this research. Anyway, that's pretty much it. And they show that this whole system seems stable and it seems to be working great, much better than noticeable improvement from the previous one and from the previous clinical trial. And I really encourage listeners to check out the paper. It's open access. It has 4 wonderful videos to show you the outcome. And it's actually incredible because there's a TED talk by Grégoire Coutine, one of the senior authors, that was shot maybe 10 years ago, where he showed some of the initial videos of his rat experiment. So remember earlier in today's talk, he had a rat model that he injected both a bit of chemicals plus electrostimulation, and the rat can move again. And the translating that to humans must have been difficult, but it doesn't look as good as the rat, perhaps, but it is significant improvement from pure paralysis. So the journey must have been incredible. And I'll upload that link to the video in the show notes just for comparison. And it's kind of inspiring that, kind of bench to bedside has occurred kind of quite so dramatically.
Great. Yeah. And you can imagine more trials, more improvements. Because, there's this wiring link. We have trouble with Wi-Fi, and you can imagine that's a big down. So it's only going to get better. This is really remarkable.
Yeah. And I think holy grail is if we can find a central pattern generator in the spinal cord that can really, by itself, cause locomotion, as opposed to kind of hacking it using stimulating the dorsal root and then of the right group of muscles at the right time. If we find the natural central pattern generator, if it exists, that would be very good to plug into.
Yeah, you're right. It is a hack, right?
Yeah, it's a beautiful hack, a kind of very good engineering hack, but still.
Wow.
Yeah.
And this I think there's been a lot of great gains in trying to figure out what the neuronal activity is actually saying for other applications, including now that there's even visual, like if you put electrodes in the visual system and then you show a scene and then you try to interpret what the visual system is actually encoding, they got to the point where they could actually just decode what the neuronal signals are and then come back and say, this is what we think the actual stimulus or what people were seeing. And I think that's just going to improve and improve.
They did it in mice too. Did you know that? It's incredible.
Yeah.
Absolutely incredible. Yeah. So I think that would be one reason why things like Neuralink, all well and good. That is kind of at least I think it's crazy that you would implant yourself so that you can surf the net without your hands. But for things like injury and stuff, I think that would really be a useful application.
Also, as they mentioned, other conditions, stroke, right?
Yeah, exactly.
Other kinds of injuries.
Exactly.
Even virus-induced paralysis, right?
Yeah.
This would potentially take care of it. It's remarkable.
Right.
Yeah.
Well, thank you, Tim. That was great.
Thanks.
That is TWiN 47. Show notes, microbe.tv/TWiN. Questions, comments, twin@microbe.tv. If you like these discussions, we'd love to have your support. Microbe.tv/contribute. Tim Chung is at New York University. Thanks, Tim.
Thanks, Vincent. Thanks, Jason. It was fun talking about this paper today.
Yeah, good job.
Jason Shepard is at the University of Utah. Jason Synaptic on Twitter, X, etc., etc. Thank you, Jason.
Thanks, Vincent.
Hey, Jason, have you moved to the new platforms, or you're still on Twitter?
Well, I think it's mostly on Twitter. I do have an account on the Blue Sky. I still find the Blue Sky a little bit more, I don't know, boring, to be honest.
Okay.
Not enough drama.
But yeah.
All right, on Blue Sky, you're Jason Synaptic also.
Yeah,
right?
same
right.
handle.
I'm Vincent Racaniello, Microbe.tv. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.